Good afternoon, everyone, and welcome to this new edition of Up Close, Stories, Objects, and Ideas from Beyond the Galleries. This is the Royal Alberta Museum's virtual talk series. My name is Eleonora Sermoneta, and I'm the adult learning programmer here at RAM. I develop programs and experiences for multiple adult audiences, and today I will be your host for this talk. And before we start, I just wanted to take a moment to respectfully acknowledge that the land upon which we stand is Treaty 6 territory. This is the traditional territory of the Cree and Nakota peoples and a homeland to the Métis. This space continues to be a gathering place for generations of Dene, Blackfoot, Tsuut'ina, Saulteaux, and Inuit peoples. We honor the cultures, languages, and histories of the indigenous peoples and this land. And please don't forget that June is Indigenous History Month, so we have a few new content on our website, and I would like you to invite you to go there and learn and explore more. All right, so thank you. During this session, we will have a presentation by one of our museum experts, followed by a live Q&A session. And if you have any questions, please type them in the chat, or you're more than welcome to use the Q&A feature. And I will be monitoring both the chat and the Q&A, so I will make sure that we are not missing any of your questions or comments. And well, today we're going to have a very interesting topic and a great talk called Piecing Together the Puzzle of Past Landscapes. So our speaker, Diana Tirlea, is going to present a fascinating topic, proxy data, which can be used as direct indicators of the past. And there are many different types of proxies, ranging from chemical, physical, and biological. And they are like these little pieces from a puzzle. So when they connect together, they can provide us with quite an accurate and interesting pictures of our past. So Diana is going to share us, her research, her work about proxy data and how they can be used to understand how the landscape, forest, and climate have changed over time. And by over time, I mean like millennia. So I also wanted to provide a quick introduction for our speaker. So Diana Tirlea has worked as Assistant Curator of Quaternary Environments at the Royal Alberta Museum since December 2012. She maintains diverse collections, including seed and pollen and the tephra reference collections. And her research primarily focuses on the reconstructions of past landscapes in the Canadian Rocky Mountains during the past 12,000 years. She also works with modern samples, including counting 1,000 of pollen grains recovered from rangeland pollinators, such as bees and flies, to better understand pollination interactions in the prairies. And aside from working in the mountains, Diana has backpacked in the mountains for over 20 years and enjoys mountain photography and painting mountains wildflowers. So thank you very much, Diana, and I'm handing the microphone over you. Give me just 1 second so I can switch the view and highlight you.
Thanks so much, Eleonora, for the introduction. And I will just share my screen now.
Oh, yes, please.
All right, so everyone should see my shared screen. Thanks so much for coming and joining us on these talks and for all the other past up close ones that Eleonora has organized. I think the introduction was really good into what I'm going to be talking about. I'm going to be talking about the past landscape and the different types of proxies we can use to understand it. All right, and I really love this picture just to kind of introduce the program I work with, which is Quaternary Environments. This is based on a project the former curator, Alberta, has done on wood bog. And it's really telling of the program because you can see those illustrations, which is done actually with one of our educators here, Nancy Nicholson. And it shows how a landscape in a certain area, a site has changed over the last 9,000 to 9,500 years ago. And how we know this is because we are finding small little remains of plants as well as animals like shells, and they each represent a type of plant or an animal, and those are indicative of a certain environment or species. So for example, over 9,000 years ago, there was wood found at the site that was beaver gnawed. So we knew that this was a prime beaver habitat, but what that requires is permanent standing water. And in modern times, there is no permanent standing water, it's more upland. So we can see those changes over that 9,000 time period. So that's what we look at. Where do we look? We primarily look at sites in Alberta and throughout Alberta, so ranging from different habitats such as boreal, prairie, and the Rockies. And we encompass the last 12,000 years primarily. And why do we do that? So even though the Quaternary is actually encompassing a larger time period, the last 2.6 million, we do the 12,000 because this is following when the ice glaciers moved out of Alberta. So about 20 to 24,000 years ago, much of Alberta was covered by these large, massive continental ice sheets, which are just massive glaciers basically. And as things warmed up with climate change, they moved out, and all these plants and animals from the south, as well as parts in the north, could move into the province. And we do this to better understand the past landscape. And it also, we can understand what kind of resources we had in the past and how changes in climate impacts those resources, the plant resources and animals. And we can use that to think about the future. If things change, what may happen to these landscapes? It's also an important indicator of biodiversity. So as our species, where they're located and what we have changes, we can also think about how the climate and environment impacted them in the past, and again, help us predict what may happen to them in the future. How do we do this? So where do we find this stuff? So these elements that we're going to talk about today are very tiny, and they are preserved in many different environments in Alberta. The primary way of getting these in Alberta is lake cores or wetland cores. But what I've been also working on is ice patches. I'm going to talk about that project today. So if you've seen the up-close presentations last year, you're going to hear a little bit about that again. But there's some other really neat places like caves, and we'll chat a little bit about a project then too. So what kind of collections does the Quaternary Environments have? So some of our collections include reference, working, archival. So I'm going to kind of dive into what the program collections are real quick, and then I'll talk about some working collections, which are the projects, to give you an idea of how these proxies can tell us about the past landscape. So our reference collections are very diverse in the Quaternary Environments, and it's not just our program that has this, it's a lot of museum programs as well as universities, research institutions. And I would have to say they're one of the most important type of collections. So why is that? Well, what is a reference collection? Well, with our reference collections, we can actually take the subfossils we find in sediment and we can actually compare them to something and confirm that they are actually belonging from that plant species or animal species. So why we can do this, especially for the last 12,000 years, is because in Alberta here, the species we've had over that time period is the same species that have occurred in North America. So we know a lot about them. So what has changed is where these species occur. Where do these plants occur? And what kind of communities and forests? Was it a closed forest or was it a prairie or a wetland at one time? So that's the kind of things that have changed. So we have a lot of information, but we do need a reference collection to understand what it is and to confirm. We have all kinds of reference collections. So a lot of the reference collections I use is the pollen reference collection and the seed and fruit. But we also have other cool reference collections like the SEM, which is the scanning electron microscope image collection, which is really close-up images of all of these cool things. And also pressed plants, which are really important. They're considered our voucher pressed plants. So they're the ones that are representing where we sampled the seed to make up our reference collection, or the pollen that we use in our reference collection. And we also have tephra reference collection. So that's all the volcanic ash. and from different parts of Alberta. So just to show you kind of just visually, we use a lot of morphological characteristics. So the shape and the different features like ridges or different sizes in some case. Color typically is not great when you're looking at subfossil because they vary, you can see that, but you can kind of get an idea here what a preserved subfossil. And the two top ones there are from ice. So the top one's from a glacier and the middle one's from an ice patch, and the bottom one there's from a cave sample. So these are spanning anywhere from 1,000 to almost 9,000 years old. And you can kind of see the reference materials, so how we can understand this. So our next collections we'll talk about is the working collection. So this is actually the current projects we have, and this is the examples I'm going to go through today. So basically working collections are comprised of like projects. So we get these samples from going in the field and collecting samples, and then we bring them back to the museum where we process the samples in the lab, and then we start doing a lot of counting, a lot of work under the microscope. Definitely to work in this kind of field, you need to enjoy laboratory work as well as enjoy working under the scope, and I do. So we have a lot of samples, and cores are a little bit of an interesting category because they span many different collections here. So there's the working collection, that's the ones we took to work on, but they also can make up the reference as well as archive components. And so what I mean when we move on to the next collection, the archive collection, is that these cores, for example, you'll see later on, you might sample some of the core and the other part of the core you will keep and preserve to have an archive of that project core. So here at the Royal Alberta Museum, for all of our programs and collections, we have special storage facilities or collection rooms, and they are special because they are maintained at a certain environment, a certain humidity and temperature, which helps preserve these long-term for decades, centuries, so that it preserves Alberta's natural history, as well as we have human history to collections in special spaces like this. And for the program I work under, we don't need that much space compared to other programs because everything is very tiny. So the pollen samples are in vials on slides, and all the macros are typically in little vials. So the largest components we have is some old pieces of wood, which I have a picture here kind of in the center bottom of some of the wood that's being found from the melting ice patches and glaciers. So overall, during this presentation, we're going to be answering the question we kind of addressed right in the beginning, how we can learn about different changes in the environment, the landscape, the forest, and the plants and animals that are within those habitats over the last 12,000 years. And the answer to this is all those preserved subfossils, which we're going to talk about, they are acting as proxies of past landscapes, environments, and climates. So I just wanted to kind of present the process for all of these. It's a very similar process for all of them. So what we do is we have some questions we're interested in answering, and then we base that on selecting the study site. So although pollen and different plant components like leaves and seeds are really well preserved in a lot of parts in Alberta, there's parts that may not preserve them as well. So like a really sandy site. Because too much water and bacterial activity is happening in there, so it degrades everything and it breaks down. So we want to make sure we select a site based on our question, but also that's actually going to have some material there. And then we go out and sample. And this is, the fieldwork is probably usually for what I do, the quickest amount of time I spend, so like no more than a week. And then you go into the last two, which is the longest amount of time that I spent. So at least 90% of my time is spent in the lab and also looking under the microscope and counting these samples. And when we process samples, we either work under a fume hood when we're using chemicals to process samples for pollen, or we do a simple wash technique using screens, which we're just running water over a series of screens with different mesh sizes to clean up the samples and then look under a microscope for those plant remains and animal remains. And yeah, so just to give you an idea of counting, right now, one of the projects I have, and we're going to talk about this on, is like you can be counting one pollen slide, like one slide in the scope, and you'll get 30,000 pollen grains. So it's a lot of time for just one sample. And for example, if I take one lake core, I might have 80 sections. That could take up to 2-3 years to finish the whole process, to process them, count them, analysis, and so on. So it is very time consuming, but you can get a lot of information from very small samples. So now we're going to move on to looking at some examples of working collections. I'll do 4 different projects I'm working on, and then we'll do questions. So the first working collection one of the most common one to get this material is lake cores. And I'm going to be reflecting back on over a decade when I was doing my master's, and I'm still working on this project because I have samples that I have different questions and more samples to count. This is beautiful Eiffel Lake near Moraine Lake in Banff. So we cored a couple of lakes. And so how we do this, so on the far left there, this is how we are coring these small alpine lakes. It's actually in the summertime. You carry up a raft in a backpack. It's about a 5-kilometer hike. And you can actually just anchor your raft, and we use a piston style core. They're a little bit shorter, and you can get your sample like that. I also have a picture here just to the right of that of winter coring. That's actually a very common way of coring in Alberta. This is often giving you, it's good for really long cores that you need. But the reason it's great is because you have a stable surface to work on, which is the ice. And then it's also helping preserve your core and sample as you're taking it out because it's cold outside. So this is a very popular way of doing it. And this is Hassey Lake, actually just near Edmonton. So, and I just took some pictures here I put up of Hassey Lake. What do you do with this core after? You cut it in half using a saw, and then you have 2 halves. And this is where it comes into those different reference collections. So you have like one side is your working collection core that you sample from, which there's a picture here, and the other one will be maintains your like archived half of the core. So you can go back to it if you have questions and so on. So what do you get? Well, for this project, I got tons of subfossil pollen, and we call it subfossil because it's not fully fossilized, like mineralized. And these are known as macrofossils. And you will see here, they all look very bright. This is not their natural coloration. During chemical processing of these samples, we add a dye so we can see it better when we're counting them under the microscope. And you can see they're really diverse, and how we can actually tell the differences between different types of pollen grain is the shapes are different. And you can see the outer edge there, the pollen wall, which is actually what's being preserved. You don't have the internal organic material left anymore after chemical processing, but that wall on the outside is really unique. You see that some are thick, some are spiky, and some have different features like net-like systems on them. And then aside from those two, you can see like you can look at shape too to give you a general idea too, which helps a little bit too. And then the pores. So on the top left there, that's alder. And you can see they have 5 pores, and there are certain ways. So pores and like colpi or like furrows cuts through them are also telling of what they are. So what can all of this pollen tell you? So it can tell you a lot of different information, and pollen analysis, the study of using pollen to answer some of these questions, is really based on questions. So some of the things I'm looking at from this project is, you'll see that picture with all that pollen film on top. A lot of us have experienced this lately or on our cars and stuff. And one of my questions is, if pollen is entering these lake systems that are quite tiny, is it impacting the animals that live in there and the algae and increasing productivity in the lake? So that's one thing it can be used to tell. Of course, another thing is looking at changes in the landscape overall. And pollen tells you one thing as a proxy. So oftentimes it's more of a regional impact, so like a regional forest. So your samples, the ones I'm looking at here, are mostly comprised of tree pollen. So I can kind of understand the flux and change in the forest from a distance. You do get some pollen in there that's from pollinators, so like insect pollinators, so you know those are more local, but that's typically pollen what you're looking at. It's not as specific to the type of species. Sometimes it's just a group. So you might just know it's pine but which pine, maybe not sure, you would base that on your site location, so on. So those are the things you can tell, and you get kind of diagrams like this. So really dense, and the top left there is the top of the core, so like today, modern, and the bottom is older, so about 2,500 for this core. And then you can look at these bars, which just show changes in a lot of the tree pollen, and you can make groupings of what may look like changes in communities. So that's how we can use the pollen to understand that. And another really important way of understanding is looking at tree line flux in the mountains. So how tree line or the forest moves upslope and downslope, which is often related to the changes in climate. So that's a lot of information you can get from just pollen. So let's look at another working collection project. This one's on ice. So if you saw last year's, this is a little bit of a recap. I am still working on this project with a really diverse, awesome group of people. And I just want to highlight it because it's really neat. It's the things we're finding. And one thing I'm really enjoying and really amazed to see is that these ice patches are acting a lot like a lake core, where they're forming these layers, and the top, near the top of the ice patch is like the top of a core, lake core. And if you're not familiar with ice patches, I can describe them, they're like baby glaciers. They're not heavy enough yet to move on their mass like glaciers do, and they often stay more still, so there's less movement, but they are typically more of a permanent ice feature in the mountains. They're down lower elevations typically too, and north facing. So why are we interested in this? Well, in the 1990s, there was a real push for ice patch research in archaeology, which was motivated by a mix of things, including climate change, which was melting some of these ice patches up north. And out of these ice patches were coming these amazing, well-preserved artifacts, like this moccasin and arrowhead. So then the research kind of got going. And this is actually, these artifacts are here is because up north, Indigenous communities, it's really important, the caribou there. And caribou go, and other also hoofed animals, go on ice patches because it helps reduce the botflies and all the other pests that are bugging them, as well as it does help to reduce their body temperature. So this is talking about summer, when they're moving up to the ice patches when it's hot, and then moving down to the valleys when it's cooler. So this is kind of why we're doing this, and we're trying to investigate in Alberta the similarities and differences. So we're looking at paleoenvironmental information too. So how are we getting the samples? So we're doing it in two ways. One way is we're taking ice blocks out of the ice patches, similar to like a lake. So the bottom would be the oldest, and near the top of the ice patch will be the youngest. And later this winter, we're also going to be doing coring. So we're going to do ice coring along these ice patches too, to get different information, as well as to line them with the ice blocks. Another thing is archaeologists are also, I'm working with them, and they're surveying these ice patches and looking for faunal remains as well as artifacts. Some of the things that have been found is this bison bone in the top middle there, as well as this leather strap. So I'm going to be talking about the paleoenvironmental aspect. So what I do is you get these ice blocks after chainsawing it, and you take it back to the lab and you just wash it down. You screen wash it with water, and that's your macrofossils, which we'll talk about what that is. And then I save the liquid and I process that for the pollen, which we already saw a bit of. So this is what we're getting from this. So we're getting a mix of microfossils and macrofossils. So we talked about microfossils, which is the subfossil pollen, and now macrofossils. So macrofossils refers to larger things. So that's the screen rostering process. So things like fruit and seeds and leaves, also animal remains, insect remains, and there's a little bit of dung in there too, which we can look at. But we also do get the pollen too. And this is really important because nowadays, this is what studies and research is leading to, is multi-proxy studies. It's very rare that I read a paper now that is single proxy, like just pollen. Even when the title is indicating it's a pollen analysis study, you kind of read the paper, and they looked at a couple macrofossils that they found in the samples to align with what the results of pollen are saying. And this is really important because each proxy is telling you something different. So for example, we talked about pollen, and how it's more regional. So it's going to give you that regional forest, looking at dynamics of forests moving upslope, downslope, whereas then you start getting macrofossils like leaves and seeds, and that's really telling of the local environment. It then even gets smaller scale down to looking at a lot of moss species, as well as orobatid mites, which there's a picture of that at the bottom right there, and that's a soil mite. Both of these can be really specific to a site, which we call microhabitats. So very specific, like, you know, a little raised ridge or something with wet moss or a stream beside it. So that could even give us a different piece of the puzzle. And each of these you kind of piece together and get a crisper picture of the changes in the landscape over time. So what can these tell us? Well, I'll just give you some examples. So we can see here we have our Arctic willow. And when we use multi-proxy approach, it also solidifies our interpretations and our results. So if I just got pollen from willow, I won't be able to tell the species of the willow from that. I can narrow down to Salix, which is the genus. So then once you start getting these capsules, which are here, they make up the floral inflorescence area, the catkin. We can see how well preserved they are, and we can actually identify those to the species. And then also the leaves, we have seeds, and we also have twigs and buds. So we put these all together to confirm it's that species. And then what can we learn from this species? Well, Arctic willow is a pioneer species. It likes to move in first, so one of the species of others. So it can help us understand the timelines for neoglacial events. So that's when ice is growing versus when it's retreating. And then this species might move in quite quickly. And then also it's kind of more of a warm liking plant. So it doesn't do well and doesn't grow a lot when there's really late snowfall, especially in spring. So it can help us understand changes in precipitation also. This plant is also important to indigenous groups. So using the young leaves as a tundra green, is also important, as well as making teas. So it can give us a lot of different information supported by multi-proxies too. So then how can we use this? So this is simplified. So I just took 3 species. The top is fir, subalpine fir, the middle is Engelmann spruce, and the bottom is our mountain, pink mountain heather. And you can kind of take those plus all the other species again in one sample, and you can build it into a community. So then you can get a picture and a visual picture, you know, using these different puzzles to understand what that landscape looked like in the past. So for example, a sample coming from an ice patch about 4,000 years ago with all these different species, and these are just a little bit of those species, there's more. Plus you put the pollen data on there with those species, and then you can kind of understand what it would have looked like. Fairly open area, very, you know, a lot of wind, pretty harsh environment, but with some trees in the near distance. So then we can understand at 4,000 years, this site actually looked very similar to what it is today. However, there was indication of tree line moving upslope or downslope at that time, and that's often driven by climate. So there's our next one, which is a little bit of a unique one. And this is Working Collections 3, looking at samples from caves. And one of the paleontologists here, Chris Jazz, is working on a lot of the faunal remains found in caves in Alberta. And so I was looking at some of the samples he obtained to see if there's any paleoenvironmental indicators and proxies that we can use to understand the landscape. So we do have caves in Alberta, some really cool ones. And some of them, like the one that we're going to be talking about, is a sub-zero cave, so you can actually see ice in it. And not only finding those bones I was telling you about to tell about what the past fauna is, there are some other things you can actually find in these caves, including the sediment, the dirt you can sample, middens, and dung. And what do I mean by middens? So here's some sampling happening of sediment. And in the middle there is an animal known as a pack rat, or a bushy-tailed wood rat. They are not a true rat, but they are a rodent. And they're the only species of pack rat in Alberta, whereas down south in the States, there's about 12, 13 of them. So these guys form something called a midden. You might have heard the word midden when you think of a red-tailed squirrel or other squirrels. They form these nice mounds in the forest, and they store their, like, you know, cache there for the winter. So all the cones and stuff, so they're good for the winter. Well, these pack rats have a different type of midden. It's actually like a junkyard midden, but it has it's their toilet too. So like their dung is in there, and they urinate, and it forms this amberite, which is like really hard and crystallized. And that's actually what protects these middens and preserves the material within it. So in the States, there's like tons of work on middens, and as well as in South America and Central America, it's like some really cool work being done. And there's limited work done on this in North America or in Alberta. So we're kind of looking and investigating if this is a useful tool in Alberta, as well as related to the environment, the caves. So what we did is they sampled a bunch of material from the middens, which was mainly consolidated debris and dung and so on. And then I put that in a bit of light acid, HCl, and kind of broke it down, and then we can actually screen it. And I also processed them for pollen. And this is what you get. So I'm just talking about two samples here. So midden 1, which is in the grayback area, is a sample from the same cave that's closer to the entrance, and it's older. It's over 9,600 years old. And then midden 2 is a little bit farther back in the cave, around 5,000 years. So they actually already have differences, even though it's from the same cave. And this difference may be for many different reasons, but it's the age is the telling one. But also we think it's not going to be just one animal. It might be multiple. We don't know. But the interesting thing is these are not just giving us information about the past landscape. It's also giving us information about the animal's diet. The thing about pack rats, they do stick fairly close to their denning area. So this is going to give us an idea of the local environment, especially from the macrofossils. For the pollen, sometimes when you're getting stuff like a lot of pine pollen and it doesn't quite make sense, it's because it may be blowing in from far distance. It's wind pollinated, and it might be landing on the material the pack rat's collecting or ingesting. So there's some differences here. So midden 1, you can see that it actually doesn't have very much material compared to midden 2. And the majority of the sample is made up of bryophytes, moss, as well as graminoid stems and bryophyte stems and twigs. And that's what you're mostly getting. And for the pollen, it's mostly actually tree pollen, fir, which is subalpine fir is in the area now. So it's telling of the forest there. And then some willow herbs. One of the common willow herbs in the alpine is called river beauty. There's a picture of it here. And there's other species too. When you look at midden 2, it's really diverse. It's not just diverse in like the different pollen that represents different plants, which we can see. It's also diverse in things like we're finding feathers, and there's lichen, and also some shrub elements like juniper leaves. So it's quite neat to see that. So what can these samples tell us about the past? So just using some pictures of the environment now, we can kind of use these to kind of understand what the landscape looked like, but also thinking about what the animal was targeting and eating or bringing into, because sometimes they do just collect and put in their midden piles. So some of these pack rats, if you're familiar with them, will steal from campsites and so on and put a part of their midden. So we can kind of reflect on what the landscape looked like 9,600 years ago versus around 5,000 years ago. All right, so onto the last working collection example. And I just want to talk about this because although the Quaternary Environments typically works on samples that are dealing with looking at the past, we also do some different projects looking at modern time. And this is important to know because those seeds, having those seed reference collections, macrofossil reference collections, and pollen reference collections can actually be applied to today's studies for modern times. So I'm working with a really awesome researcher at U of A, Carol Frost, and she's an ecologist, entomologist. Just a little bit of a thing here. I'm not an entomologist, so I'll try and answer all the questions you have, but related to insects. We'll see if I can actually get those answered or not. But I'm doing the pollen component. So we're kind of collaborating on this. She knows the bugs and insects, and I know the pollen stuff. So this site is out in the prairies, in the rangeland, and it is looking at the different pollinators, both native and introduced, such as honeybees, and seeing how they can, what kind of plants they're going to and what they could be pollinating, and if they impact each other's area that they're going to. So how do we get these samples? So my primary role in this is looking at the pollen. So the students that work with Curator of Frost actually collected the samples, and they are doing other studies on the pollinators. And what's important to know here is if we're interested in the pollen that is actually going to be a source of pollination to another plant, we're interested actually in the pollen that's on the body of the insect. So not the pollen baskets. So that's the ones, if you see a bee landing and collecting pollen, you'll see a thick yellow band on their hind legs. That's what they're collecting for food. So we don't want to use those. So we're just sampling the pollen from their body. So these were collected and processed by the students too. So I've been making slides. I have reference material also that I have from the plants that are in the area, so voucher specimens, and then I can use those and start counting. So what are we finding? Well, we're finding a lot of cool pollen. All of these have been colored, so you can see they're quite bright pink. They're really neat. But in the prairies, we have some really interesting plants like pincushion cactus and evening primrose. They're beautiful. And what we're finding is something really interesting. So right now I have over 200 samples to count, and I've done about 50. So there's still a lot to look at, but we are seeing quite a bit. So anywhere, I said in the beginning, around 20,000, 30,000 is what I'm counting per slide. So it's a lot of pollen. So let's see what this information is telling us so far. Again, we're still working on this project. And as you count and add more data, the information might get more specific and crisp, and you'll understand it better. But so far, what's happening is the majority of the samples that I've counted are kind of like the one on the right. So what happens is a pollinator lands on a flower, a type of plant like say this chickpea milkvetch, and then they are captured and sampled there. So when I look at the pollen slide from that species, that's been the specimen that's been collected, the majority of the pollen is actually coming from what it's been captured on. And then there's a smaller component from different plants around. So that's saying that the primary source of pollination is going to be for the same species. Whereas there is a few, and I mean, this number might broaden and like expand as I get through all the samples, but there's some interesting ones where the one on the left, the honeybee that was captured on this bellflower, actually didn't have bellflower pollen. So we don't quite know why yet, but that maybe there's so much pollen on the body already, or maybe that flower was not producing pollen. That would be a good explanation at the time, because different plants produce pollen at different times too. It's not all in once. As you kind of know by right now, the pollen release is all our tree pollen, our spruce and so on. So yeah, so there's two different pictures here that we're seeing. And now we also need to apply some statistics to see if there's any competition between the different pollinators too. And that will be Carol doing that kind of work. So a lot of people ask me who can use these collections. And at the museum, if you're interested in using a collection for research or studying or looking at, you can reach out to the people that are working with them. So I just have my email out here. So for example, the kind of individuals that use the Quaternary Environments collections includes, we have a lot of students, professors. They might come and look at the reference collections to compare them to samples they've collected on their own site. We're also having people who just send me requests like, hey, do you know what this is? This is the project I'm working on. And also people such as artists. So artists have reached out and said, hey, I'd really like to do this collage or this piece I'm working on. Can you provide me images, or can I come look at the collection? So it's really diverse. I also have a lot of volunteers working. You can apply to volunteer at the museum through the website. And I have volunteers working, which also makes use of these collections as they help accession the collections and so on. And every collection is different, so you just have to reach out to people and see what's possible. But yeah, please do reach out if you're interested in knowing more too. And I just wanted to end the talk with some resources. If you're really interested in learning more about Alberta's plants, as well as the seeds and the fruits, there isn't a real specific guide to seeds, fruits, and pollen for Alberta specifically. It's kind of a mixed match. And a lot of these guides here that I have actually have descriptions of the fruit and seeds. So for example, sedges, carex, that's the way you're going to identify them and confirm ID. So it actually has really good descriptions in a lot of floras. One of the best online floras for Alberta is Flora of North America, and also eFlora of BC, which I didn't list here, but they have very similar species that overlap, especially the mountain area. And you can go there and it has images and descriptions and ranges and different uses, like medicinal uses and so on. So it's a really helpful website. They don't have it quite completed yet. They're transferring the book, information into an online system, but it's a great resource. And there's some other things here too. Nature Links and iNaturalist are great. They often have areas you can search, like Jasper on iNaturalist. They have, if you're going to Jasper and interested in the flora there, you can search Jasper flora, and they actually already have made lists that people have kind of contributed to taking pictures and stuff like that. And you can learn your plants like that too. And again, you can email me if you have any questions on resources. These are just a very tiny bit. And the Rare Vascular Plants of Alberta has just come out. So awesome. And we have one of our assistant curators of botany contributing to this, Donna. So very cool book. And the White Horse Wildland, if you've never been there, fantastic place to go to. So I think I'm done. I'm done a little bit early. So yeah, I'll stop sharing. And if there's any questions, Eleonora will lead that.
Thank you very much, Diana. That was a really fascinating presentation. Thank you for sharing your work with us.
Thank you.
And yeah, we're now moving to the next part of our program. So we're opening the floor to your questions, and please feel free to use either the chat or the Q&A. Again, I will be monitoring both of them, so I'm making sure that we're not skipping any of your interesting questions. Let's give it maybe a moment. Because yeah, I know that you folks are typing the questions. That's maybe not ideal, but in this way, we make sure that we capture all of them. And we have a comment from Brian. So Brian says, excellent presentation. Thank you very much. I'm glad that Brian enjoyed the presentation. Okay, so we have a question. So, hi, thank you for the presentation. Your job looks really exciting. What's the aspect you enjoy the most?
Oh, thanks so much for that question. Yeah, I think the aspect I enjoy my job the most is actually the diversity in it. So I really enjoy, although I'm in the lab quite a bit, I really enjoy being able to, do presentations like this, but also go in the field. I have the privilege of being able to go in a helicopter to get to my site. So I get a lot of interesting opportunities and meet a lot of great, interesting, diverse people that I get to work with.
Thank you so much, Diana. I actually have a question for you, because you get to explore many different places and collect samples, in different areas of the province. So, do you have a favorite spot for your collection?
Yes, I think it's the most recent project I've been working on. The Ice Patch project is working in Jasper, and it's very close to Cavell area. And it's just a wonderful place to be, working, collecting samples, doing surveys of the plants. And I love getting down on the ground and finding all those little treasures. There's a lot of tiny plants in the alpine that are just beautiful. So yeah, it's probably one of my favorites.
Thank you for sharing that. And more questions are coming. So, oh, this one is pretty interesting. Are the snow patches fragile? Is there a risk of damaging them while digging for fossils?
Yeah, so what we're doing, the risk is not high compared to what's happening with climate change. So some of these ice patches may be gone. It could be rapid, like 10 years, but for sure by 50 years. So the importance is that there would be high damage to any artifacts that are released from these ice patches. So especially up in the Yukon, when I was talking about that project, they're surveying every year. They have Indigenous communities, they work with them, they survey every year, because as soon as a artifact or a wood, so we found a lot of wood from this ice patch that are dated to almost 7,000 years, which tells us when the ice patch is formed at minimum age. And once they're out of that ice, they just degrade very quickly and can get lost. So yeah, I don't, based on what we're doing, the precipitation of snow is gonna, we can't see those blocks when we go the next year to look. So it's all been covered and stuff, but what we can see is meters of melt around the edge. They're rapidly disappearing. Yeah, good question, yeah.
Thank you. And well, this one is another interesting one. How can I get a similar job? Maybe you're inspiring the next generation of researchers here.
Yeah, that's a really good question. Yeah. So just from my background, where I got is you typically need a degree, a master's or PhD. I did the biological or ecology route, but a lot of individuals do the geology route. So that's for my position specifically, but also there's a lot of technician positions that go out and survey and do similar work, and that might require an undergrad degree at a university. And I always say this, volunteer, call people, talk to people. That's where your passions will come out. And before this, I actually wanted to be a veterinarian. So my course and career changed drastically after I worked and volunteered and talked to people to know what I would like the most.
Thank you. And yes, we have a volunteer program. So if you're interested in spending some time with us, exploring the collections and learning from the best museum professionals that we have here at RAM, just get in contact with us. All right, and I actually have another question from the chat. So I would like to share this presentation with my friends at the university. Oh, sorry, I lost that for a moment. Are recordings going to be available online? If yes, where? Oh, okay, I guess that I can respond to this question. So yes, we actually have recordings for all the previous talks from the Up Close series. Just give me 1 second and I will share that link with you. So they are in on the same web page where you registered for this talk. And if you scroll down towards the bottom of the page, you will find all the previous recordings, and the specific recordings for this talk, they will be available towards the end of the next week. So just keep an eye on that, and yeah, make sure to check the page regularly. Okay, I have a comment from another visitor. I must leave. Thank you for the presentation. Love your enthusiasm for native plants. So thank you. Thank you. It's great to have your feedback. By the way, I shared the Royal Alberta Museum's webpage in the chat. Let me know if you're having trouble with opening the page, but that should be quite all right. Okay, let's see if we have other questions. Okay, I have one more. Oh, what's your favorite plant, either for research purposes or for your backyard?
Oh, for your back? Oh, that's awesome. I actually just planted some mountain avens, and it's been doing really good. I live in Edmonton, so I put it on a dry slope, and it's also known as 8-petal avens in the past. It's a white flower, and it's doing amazing. So I really recommend that. And you might know prairie crocus for the garden, or pasque flower is really good. But those are some plants that are doing great, and I love my garden. But my favorite plants are actually, I named my kids off of them. So Cassiope mertansiana, which is a mountain heather, it's very tiny, low growing. And then Linnea borealis, which is twin flower. And it's a very cool plant. If you don't know Linnea borealis or twin flower, look it up. It has one stem and two little flowers come off the same stem and they kind of hang down. So those are my two favorite plants.
And one more question. Where do you conserve the artifacts you find in the snow patches? Are they visible at the museum?
That's so awesome. So the actual ice with the samples or before processing, we have freezer units that are collection units. And the actual samples that I cleaned and processed and are in vials are in the collection rooms. Nothing's on display right now, but there's hopes to have a mini exhibit on this project because it's quite getting larger and larger with more and more people. And also to say that if we do back of house tours too. So if you contact me and we can discuss and see if that's possible. And then you can see a little glimpse of that because all the samples are currently in the back of house right now.
Yes, exactly. And, you know, here at RAMA, we have 2.5 million objects, you know, that we conserve. That means that we have 2.5 million of stories to share with you. Of course, we have a back of the house section where, you know, we store and keep a multitude of specimens and objects. And yeah, so just a small, tiny part is actually, you know, on display in our galleries. But yeah, just send us your questions, inquiries, and, you know, we will make sure that you are put in contact with the right people here. Okay, we have a few minutes left. Just let us know if you have more questions or even, you know, comment, feedback, or maybe your experience in the mountains as well, because we all have something to contribute. Or maybe if you're planning a nice trip, you know, in a mountain area, now you have more information about plants and maybe you will be more aware of your surrounding. So yeah, I find, you know, what we do here also an important part of how we experience our, you know, our landscapes, our mountains. So yeah, if you are interested in more stories that are related to the past of the landscape, make sure to take a trip here at the museum. We have a wonderful natural history gallery where you will find more stories and experiences. And I have a feedback and a question for you. So, hello, thanks for the presentation. That was really interesting. Is it possible to visit the museum storage like with a guided tour? Who's gonna take this one? I guess we can.
You want to start?
Yeah, sure. Yeah, again, just make sure to contact us. You can contact us through our general inquiry form on our website, and we will shortly, you know, get in contact with you and address your questions. I'm sure that we can find a way to provide, you know, some experiences to you. But yeah, obviously, we need, you know, sometimes to process those requests. So yeah, just get in contact with us.
Yeah, and thank you for the comment. Yeah, I've done back of house tours arranged through Learning and others, so like for the collections. So yeah, just reach out and see what's possible.
Oh yeah, definitely. Yes, of course. You know, or we have a different process for back of the house tours. So clearly, we need some time, and again, we need to make sure that we have the capacity to do that. Yeah, but that's actually a very interesting question because yeah, so we have so many stories to share, and you know, sometimes some of them are right at the back of the house. They are just waiting to be shared and discovered, you know, with our visitors. All right, let's give it one more minute. Yeah, this was a really great presentation, Diana. Thank you for that. I guess lots of the research that we do here, sometimes that remains within the walls of the museum, or, it is shared just with some specialized community of interest. So what we want to do through Up Close is to bring all those stories, all that fantastic research that our amazing curatorial team does. And, we want to share that with our visitors, because this is what museums are for. We're all about sharing stories and reaching out to our diverse communities. And I actually have another question for you. So can you remind us what your education background is? I guess that there is lots of interest for that. Hey, Diana?
Yeah, I usually get a lot of questions, and I actually just attended a career week at a school this week talking about it. Yeah, so I don't know if you want it, but my background is I have an undergrad in zoology because I wanted to do my veterinarian. And then I changed my mind and I did a master's in ecology. So even though I'm looking at changes in the past, for my degree in my master's, I looked at lake cores, the Eiffel, the first project I went over, Eiffel Lake and Sentinel Lake. And I looked at changes in the pollen, so which represents the vegetation, along with changes in algae, so the productivity. So that's my background.
That's fascinating. Thanks again.
Yeah.
Yeah, I'm so amazed because, we're getting all these questions. Sometimes we get very technical questions. And this is the first time that we're actually getting specific questions about your background, your studies. So this means that you're having an impact and inspiring, new researchers to join this amazing field. So thank you for that. And I guess we don't have any additional questions. So I just wanted to thank you, Diana, for sharing these stories with us. And thank you, everyone, for joining this last talk of the Up Close series. So I hope you enjoyed our presentation. And, before we say goodbye, I just wanted to tell you a couple of things. We have a super quick program survey available at the end of the talk. So obviously, we want to hear from you, because as I told you, museums are all about engaging with diverse audiences, and we would like to learn what kind of programs and experiences you would like to see here at RAM in the future. So this super quick survey will open automatically in your browser at the end of this talk, and the data will be collected anonymously, of course. So feel free to let us know how you felt about the presentation, the program in general, what kind of things you would like to see here. And if you don't want to participate in the survey, that's just fine. You can just close your browser. So this was our last talk for the year. Thank you, everyone, for attending Up Close, stories, objects, and ideas from beyond the galleries.
Thank you, everyone. Thank you, Eleanor.
Thank you, Diana. Thanks a lot.
Bye-bye.
Bye, everyone.